Alright guys, we are going to finish up our DNA technology lesson, and afterward you're going to do a little practice quiz. It's just to kind of expose you to some questions that you might see on the test. Since we don't have an actual quiz and it's been quite a while since we started the notes, it's just a way to help you practice. So we left off talking about vectors, and so I just want to do a quick recap on those because this whole plasmid concept is really important to understand. Remember that a vector is just a vehicle. It's a means of transferring something from one place to another, and in this case, we are transferring genes. And so one of the most common vectors is a plasmid, those little small circular pieces of DNA. Tomorrow you guys are going to be doing a paper plasmid activity, and we're going to create a recombinant plasmid, and that's an example of something that might be used as a vector to insert a gene into a bacteria. Now, another common vector were phages. Remember that these are bacteriophages, or viruses that infect bacteria, and so they could transfer genetic material by using one of those viruses as well. And so once, remember that we get the gene into a bacteria, the bacteria can then just kind of go to town, and it can make copy after copy after copy after copy, and that's what we call molecular cloning. It's a way that we can copy genes using bacteria cells. Now, another thing we can do is once we have these genes kind of isolated in these bacteria, we can then create DNA libraries. And so think about these bacteria like books in your library. Each bacteria group could contain a different gene. And so that way, if you wanted to use them for genetic study, the genes are already isolated. We already have specimens that contain them, and then those bacteria could be used to study those genes. Now, if it was an organism where we've mapped out the entire genome, you might have a library that contains every gene for that organism individually located in bacteria. And so these DNA fragments are in the vectors, they're put into the bacteria, and then they can be stored to be used for later study. Now, when we create these DNA libraries, often we create something called cDNA. And the C stands for complementary DNA. Now, this is going to take us back to first semester for a moment, and I want to talk about transcription. Okay, so remember that in the process of transcription, we start with DNA, and then DNA is converted into mRNA. I really don't like this. All right, so we have DNA to mRNA. That is the process of transcription. Now, I hope you remember that whenever we copy the DNA, we copy the whole sequence from promoter to terminator, and that often includes these little sequences called introns. And remember, we considered the introns to kind of be the junk in the DNA sequence. Okay, so this original mRNA here, this is what we call our pre-RNA or our primary transcript. This original mRNA contains both the introns and the exons. And if you remember, the mRNA gets edited. Those spliceosomes come in, they cut out the introns, they piece back together the exons. And so what's going to happen is the mRNA gets edited. And so after the process of editing, that mRNA has exons only. Sorry for the clicking. Well, that was useless. All right, so this mRNA only has exons, okay? Now, if I want to store the genes, if I want to store the genetic information, I would much rather store it in a form that is already ready to go, okay? It's already been edited. And so what will be done is this mRNA that has been edited, that is exons only, will be turned back in to DNA, okay? Now, that is going to require an enzyme known as reverse transcriptase. And we actually have seen this already. Back in our virus notes, we saw that retroviruses have reverse transcriptase. And what it does is it converts RNA back into DNA. So we started with DNA, we turned it into mRNA. The mRNA got edited so that we only have exons. And then we turn this edited RNA back into DNA. And this is what we call cDNA, okay, or complementary DNA. So now what we have is the DNA gene that has exons only, and now it's ready to be placed into a bacteria, and it can more easily be expressed because the junk information has been removed, okay? So cDNA is just DNA that has been edited, okay? And it was made from mRNA, and it requires that reverse transcriptase enzyme. So when we're creating these DNA libraries, we typically store it in the form of cDNA. So once again, you take the mRNA is converted back into DNA, and then this is the DNA that we put into our vectors to house our genes in our library. All right, so far we've talked about molecular cloning, and that was just cloning genes. But we're also going to take a look at reproductive cloning, and this is where you make a whole organism. Normally when we think about cloning, this is what people may think about. And this involves parthenogenesis, which is when you can create an embryo without fertilization. So we create an embryo without egg and sperm coming together. And so essentially what you do is you take an egg, okay, an egg cell. Now, normally an egg cell would be haploid. It would have a half set of chromosomes from the mother. And so what you do is you take that egg and you remove the nucleus, okay? So you just take the nucleus out completely and you replace it with a diploid nucleus. So let's say that I wanted to clone myself, okay? You could take an egg, you remove the nucleus, and then you could take a nucleus from one of my cells, let's say a skin cell that's a diploid cell, and I could put that nucleus into the egg. So now it has the 46 chromosomes. It doesn't need to be fertilized. It already has the 46 chromosome count. And so that DNA is specifically my DNA. Now, just like any other organism, that egg would have to go through development, and it would have to divide, and it would have to differentiate, and it would have to grow into the embryo all the way into the fetus and the baby and all of that good stuff. So if I wanted to clone myself, it's not like I could clone a 43-year-old me, okay? It would have to go through the growth processes. But if that egg contained my full genetic instruction, then it would grow up and it would have the same genetic composition as me. So the egg would be placed into a surrogate, and it would develop like any other organism, and then when that baby was born, it would be a genetic clone of me. And so this kind of was first done with a sheep. At least I think it was a sheep that was first. There could have been some other organisms that they cloned. But I guess one of the most notable, the first mammal that they cloned was a sheep. And so they took the nucleus from a grown, fully developed sheep. They placed it into the empty egg. They put it into a surrogate. And what you get is this baby lamb that is genetically identical to the original sheep. Now, this particular lamb was named Dolly, so it's kind of notable. Now, the thing is with these clones is a lot of times they do have abnormalities, because if you think about taking a nucleus from an adult cell, that nucleus has been exposed to things. It could have accumulated mutations, so it has been around longer. So if you think about things like those telomeres that we talked about, it's already been copied several times. And so that DNA is not the same as a freshly produced zygote, okay? And so there could be some issues with that DNA. And so typically the clones don't live very long. A lot of times they are more susceptible to certain diseases. Now, there have been attempts to clone human embryos. This has definitely been a source of controversy because there's disagreement on where life begins, and so some people say it's okay, and some people say that it's not. But embryos are a pretty good source of stem cells that can be used for all different types of purposes. And so when it comes to cloning humans, it's pretty much considered not okay by the majority. And so we really shouldn't be cloning humans. And really the thing is even if you make a genetic copy of something, it doesn't mean it's the same thing, okay? Like people are cloning their pets and things of that nature. Well, part of what makes us who we are is the environment that we grow up in and the experiences that we have. So even if I were to make a clone of myself, or if you were to make a clone of your dog, keep in mind that it's not, while it may be genetically the same, there are other factors that make us who we are. Now, another common practice are GMOs, and this actually started way back, like in the 70s. If you remember the video that you watched, this genetic modification started a long time ago. A lot of people are still scared of it, but really all you're doing is putting DNA from one organism into another. It hasn't really proven to be specifically dangerous. Now, there could be some long-term environmental ramifications, and part of it is we just don't know what the long-term effects are, and I think that's why some people are afraid of it. But really, you're just transferring DNA from one organism to another. And so it starts by making that recombinant DNA molecule. This is what we're going to do in our paper plasmids. And then as soon as you introduce a gene from one organism into a new organism, it is now genetically modified, okay? And we call that organism transgenic, okay? So we've been transforming and turning these bacteria and plants and genetically modifying animals for quite a while. And the goal is to make them better, like in terms of plants and animals, make them more resistant to disease, higher yield, maybe better meat, whatever it is we're using them for. And it has been around for quite a long time. We also saw in the video that we like genetically engineered bacteria to clean up oil spills. So there's all different types of things that we can do. It just simply involves putting a gene from one organism into another. Now, in order to do that, it involves the process of transformation. Now, we saw transformation back in the bacterial genome notes. Transformation, by definition, is just the uptake of foreign DNA, okay? So if we're transforming an organism, we're trying to get it to take in the foreign DNA. And so what we can do is we can kind of play with those organisms like the bacteria. We can expose them to certain conditions and make them more susceptible to taking in that genetic information. So if we transform an organism, we just transfer that gene into them, okay? Now, back when we were covering DNA, we talked about Griffith's experiment. It was the transformation experiment where he had like the live pneumonia bacteria and the dead pneumonia bacteria, and somehow the dead ones transformed the live ones into the bad strain. So we know that bacteria can just naturally take in genetic material from their surroundings, but we kind of take advantage of that and try to force that transformation to occur. Now, once again, we need a vehicle, and so typically we use either a plasmid or those bacteriophages, and it's just a way of inserting a gene into something else. This is what Boyer and Cohen did from the video. They first made a recombinant plasmid, and then they got that plasmid into a bacteria, and they created that bacteria that made toad proteins. And then we saw that carried forward by creating bacteria that can make the human protein insulin or human growth factor or other medications that could be beneficial to us. So we usually start with the plasmid. Remember that that plasmid needs to have the origin of replication so that it can be copied. We also need a selectable marker. Remember that we typically use antibiotic resistance. This is going to help us determine which bacteria actually got the gene, and that shows us that they actually were transformed. And so we used to do a lab in AP Bio. It was a green gene lab, and so we were able to transform bacteria. And if we knew we were successful, if they grew in the presence of antibiotics, and they normally had a greenish color. These are some other animals. They've been transformed with glow genes, and you can see that clearly they have some new traits that they may not have had before. And so pretty much almost any gene can be inserted into bacteria, or sometimes we use yeast cells because they're unicellular and copy a lot, just like the bacteria. And so it allows us to produce large amounts of protein product. And so this has really revolutionized pharmaceuticals. It just needs expression vectors so that they can actually express the genes. Because remember, just because you have a gene doesn't mean you express it. And so this is like what we saw in the video, using those bacteria to make human proteins that can help in medicine. All right, some other advancements that we've seen is genome mapping. Now, I'll be completely honest and say that I don't fully understand all of this. A lot of this is computer generated. They're able to put the information into computers, and then the computers do all the work. And that's well beyond my knowledge because I do not know all the technology side of it. But essentially, we can map out our genes and find out what chromosomes they're on, where they're located specifically, and then we can also create maps that show the location of all the different genes. And all of this data is going to be stored in the computers on databases, and everybody can kind of access them. And in terms of the human genome, we're still learning and identifying every single gene and exactly what they do. So one of the ways we can study kind of the gene function is with the genome sequences and these microarrays. So we can sequence them, which just means literally figuring out the letters that make up the code, like ATTCCG. And once you sequence them, you can identify the genes, and then you can study those genes to determine what they do. We can also study genes in different cells to see which genes are active in which cells and when are they inactive. We can compare cancer cells to normal cells, and this is often accomplished with these microarrays. And so they take these little tiny pieces of DNA and they put them like on a little computer chip. It's a little tiny chip, and they've got lots and lots and lots of DNA fragments on them. And then they find the mRNA. They take mRNA and they try to determine which genes are being converted into mRNA. Because if they're being transcribed, then they are active. If they're not being transcribed, then they're inactive. And so it looks something like this. Okay, they take the mRNA, they convert it into the cDNA, and then that cDNA will then kind of attach to those DNA sequences. And so if they're matching up, that means that the mRNA was made, and that means that those genes are active. So here they're comparing normal cells with disease cells, and they can see which genes are turned on in both, or which genes are turned on in only one. So mainly you just need to understand it's just a way to analyze and look at gene function and gene regulation. And so we can also do that by kind of manipulating the DNA. We can kind of create mutations and see what they affect. Now, we don't really want to do this in humans because we don't want to intentionally create diseases, but they can do like knockout experiments with mice where they kind of turn the gene off. They can block transcription, or they can block translation with things like antisense RNA or this RNA interference. And so by blocking the transcription or translation, they can see what effect that has on the organism, or what protein is not made, or what pathway is affected. And so this is another way that we can study genes and try to determine exactly what those genes do. Now, we've talked about a lot of different technologies, and to be honest, most of these technologies have been around for a while. I'm sure there's a lot of advancements that have taken place since this PowerPoint was made. And like I mentioned in the very beginning, hopefully maybe some of you guys will go into this field and maybe even be a part of some of these new technologies. But even with what we do have today and what we've talked about, we know that there's a lot of really beneficial outcomes, things like diagnosing disease, creating those pharmaceuticals. Now, gene therapy is not something that we do on the regular yet, but it is a potential benefit, and that there could come a day where we could possibly cure some genetic disorders. Most genetic disorders have treatments but no cure, because it's kind of hard to go in and change the genetics of every cell in a person's body. But if they're just simply lacking a protein or something like that, with gene therapy, there's potential to potentially cure those diseases. So this is not something that we do on the regular. We try not to manipulate human DNA too terribly much, but there are some, I think, trial runs going for certain diseases, and this could be something that we see on the regular in the future. We've been using it for forensic study for quite a while, things like PCR and DNA analysis, DNA fingerprinting, the environmental issues like cleaning up oil spills or cleaning up toxic waste. And we definitely saw how it can be used in agriculture, like with Rob Horsch, and creating disease-resistant crops, pest-resistant crops. And so these are just a few. You may read about some more when you do your study guide. Hopefully you're working on that. But this is going to conclude your DNA technology notes. I'll be seeing you guys soon for the paper plasmid lab. I hope you guys have a great day.